Our next presenter is Stephen Spelunky from the USDA.
All right. Hi, everyone. My name is Stephen Sapolsky, and I am with the USDA APHIS out of the Forest Pest Methods Lab in Buzzards Bay, Massachusetts. And today I'm going to be talking about the rearing, life history evaluations, and host range testing of Dryaena sinicus, which is proving to be a promising biocontrol agent for the spotted lanternfly. So I just want to start off by saying thanks to all of our various collaborators. This really is a collaborative effort that I'll be presenting today. We have a great team of collaborators both across the US and in many countries abroad. So their contributions are really what's allowing all this work to get done. So just a very brief overview of what our group has been involved in at the Forest Pest Methods Lab as far as spotted lanternfly biocontrol. Our group really began our work in 2015, and among the first things we did was we did some foreign exploration where we were able to identify two natural enemies of the spotted lanternfly in their native range in China. And the first of those is an egg parasitoid, Anastatis orientalis, pictured on the top. And the second is Dryaena sinicus, which is a nymphal parasitoid, which we'll be focusing on today. After we identified these with the foreign exploration, the next few years of our work was really heavily focused on that egg parasitoid Anastatis orientalis. We found a lot of ways to really get in depth with research on that wasp, and we spent a lot of time looking into a lot of ways to optimize rearing strategies as well as getting continuous colonies. But we really dug into a lot of life cycle and life history studies with the Anastatis orientalis wasp as well. And so we were able to do things like longevity, fecundity. We did some genetic analysis where we were able to identify 3 separate haplotypes of that Anastatis wasp, and we were able to put those all through testing individually as well. And finally, we got into the host range testing of that Anastatis orientalis. And at that point, we did discover, unfortunately, that all of the various haplotypes that we worked with did have too broad of a host range for us to continue our work with that parasitoid. And in particular, we did find that all of those haplotypes were really prone to attacking non-targets such as silk moth eggs. So at that point, we really started shifting our focus over to Dryaena sinicus, that nymphal parasitoid, which is what we will be focusing on today. So Dryaena sinicus is a wasp that is native to China alongside the spotted lanternfly. It's a univoltine nymphal parasitoid that attacks first, second, and third instar spotted lanternflies. And we found that they have a particular preference for first and second instars. The females will use their long raptorial front legs to hold on to their prey, and they'll do this both to host feed as well as parasitize the nymphs under the wing pad. And then after parasitism, a larva will develop inside what's called a thylacium, and that's really just a protective sac that grows on the outside of the nymph's body. And then after development, the larva will burst out of that sac and kill the nymph, and then the larva will go on to spin a cocoon. So there's a variety of factors that really went into why we're interested in working with a dryaenid wasp as a potential biocontrol candidate. So we've seen that they're really highly effective in China at parasitizing spotted lanternflies. And in some cases, we've seen parasitism rates of up to 60% in certain field sites. As I described earlier, unfortunately, our other option for a biocontrol candidate that we know of right now, that egg parasitoid Anastatis orientalis, unfortunately did show low host specificity and is likely unsuitable for further work and release. But however, dryanids have been successfully released around the world to control invasive hoppers before. And in particular, one of the better documented of these is the result of the wasp Neodryaenus typhlocybe to control the invasive plant hopper Metcalfa pyrenosa across Europe. And studies for this actually showed when they looked at the host range testing for this wasp, there was no non-target attack across any of the 8 native hopper species that they did end up testing. And follow-up studies showed that this wasp had pretty good establishment in surrounding countries that were also infested with this hopper, but didn't do any of their own releases. And in fact, after just 4 years past the first detection of this wasp, it was found at 38 out of 39 locations with parasitism rates up to 30%. And then finally, we've also just seen observations of native dryinids a little closer to home being found as our collaborators at URI were collecting native hoppers for our non-target testing. They were finding those to be parasitized fairly regularly by native dryinids as well. So all of this is just kind of adding up to us seeing that dryinids are quite effective parasitoids no matter where you find them in the world. The life cycle of Dryonis is also something that lines up really well with that of the spotted lanternfly. You can see that the vast majority of the life cycle of this wasp is spent overwintering in that cocoon stage, but it's pretty clear that their seasonal reproductive period lines up really well with when they'd be encountering the spotted lanternfly nymphs out in the field in that late spring through the summer. So they're able to naturally encounter their prey. However, for our rearing purposes, we actually need to attempt to kind of override this natural drynid cycle so we can be continuously producing wasps and storing those cocoons all year so that we can kind of build up a stockpile so that we'd be ready for a single release event in June when they'd be able to go out and encounter those mid-instar spotted lanternfly nymphs in the field. So in order to kind of tackle this, we developed a multifaceted rearing strategy, which kind of brings a lot of moving parts together all at once. So the first part of this is growing our host plants. So we will go out, we'll collect and cold stratify tree of heaven seeds in the late fall. And then after they're cold stratified, we'll propagate those and maintain those in our greenhouses until the trees are the proper size to be used in the cages in our studies. And then kind of at the same time that all this is going on, we'll also go out and we'll collect spotted lanternfly egg masses from the field. I was actually in Pittsburgh yesterday doing this, and then now I'm here talking about it. But yeah, so we'll collect those spotted lanternfly egg masses from the field, and we'll bring those back to the lab. We'll store those at 5 degrees Celsius, and then we'll bring those egg masses out of chill and we'll warm them up periodically throughout the season so that we can continue to produce nymphs all year round so that we can feed our wasps. So the third part is obviously rearing the wasps themselves. So when we get those cocoons, those will overwinter at 5C as well. And then we'll warm up those cocoons at what we call Beijing spring conditions. And this is really just a set of environmental parameters that kind of mimic the temperature and day length of what Beijing would be experiencing through like May and into June when these wasps would naturally be emerging. And then after the wasps do emerge, we'll hold males and females together for mating before they go into their oviposition period. And all three of these processes will align so that we have our desired second instar spotted lanternfly nymphs in our cages with some mated female wasps ready to parasitize them. And then the product of that is those parasitized nymphs will have the Thylacia develop on them for about 3 to 5 weeks, after which the larva will emerge out and produce those cocoons, which we can harvest and then store those or try and rear those out again and move into our various colony building and studies. So we entered 2023 with a handful of goals to further our work. And today I'll kind of be running through the progress we've made on each of these and where we are now. So for the first goal of maintaining a strengthened and continuous colony of Dryonis indicus, coming into 2023, we were able to rear spotted lanternfly nymphs and wasps from about February to September, which is actually quite a bit better than we were actually even able to do 1 year prior. And what we were seeing in coming into 2023 is that when we produced these cocoons, we would store them at what we're calling Beijing match conditions, which is just, again, a set of parameters that are mimicking kind of in real time conditions like the temperature and lighting of what these cocoons would be experiencing in the field in Beijing. And what we saw is that about 15% of the cocoons that we produced would actually develop and emerge before going into any chill period, which is not what we expected. But it did allow us to have a few wasps kind of at all times with this trickle emergence. So we were able to, kind of continue building some colony and continue work, but we weren't working at like true colony numbers because this was not the ideal rearing scheme for these wasps. And we also were still kind of playing around with a consistent warming scheme for these cocoons once they were coming out of chill. So we weren't sure what would give us the best hatch. So in an attempt to kind of build ourselves further towards a continuous colony of Dryonis indicus, we made some adjustments or additions to our rearing strategy. So as we were producing these cocoons throughout the season, when we would produce cocoons earlier in the season, like March through June, we'd take these cocoons and we'd chill them down at 5C for a month or two before warming them up in those Beijing spring conditions, which we did end up finding gave us like our best hatch rate of like over 50%, which still isn't ideal. Our collaborators in China are able to have up to like 70 or 80%. But so we're still working towards a better hatch rate. But for us at this point, this is how we can get our best consistent hatch. So this allowed us to have more wasps to work with because there was a higher hatch rate than that trickle emergence, 15%. And it also allowed us to have additional generations that we were producing in a single year. However, when we produce cocoons kind of later in the season, those we would actually put back into those Beijing match conditions so that those would only have that little 15% trickle emergence. We'd still be able to have some wasps to work with later in the fall, but the majority of those wasps would not develop, and those would go into the fall and they'd go into a very natural chill in November. And then, so they would overwinter at 5C. And then now we are bringing those out of chill right about now and warming those up again in the Beijing spring conditions for our 2024 work. So we've also made really good progress as far as building a strengthened colony of Dryaenus sinicus. This graph shows our colony this year in comparison to a couple of years ago as well. So in 2021, we were only able to produce about 95 cocoons in total, and those were all male because we had not figured out how to successfully mate these wasps yet. And then in 2022, we were able to produce 775 cocoons and produce females for the first time. And then this past year in 2023, we were able to produce over 2,100 cocoons with a lot more females to work with than even in 2022. So we are definitely making good progress on that front. Another thing we were able to accomplish in 2023 was very early on preliminary genetic testing just to see what we were working with as far as the wasps that we had. Two wasps did end up kind of sequencing out to be highly different from the other ones, about 3 or 4% difference. And we think that these are actually potentially could be like a cryptic species that was given to us from those field collections. But the rest of the wasps that we did test kind of broke nicely into 3 haplotypes. And those were pretty kind of ubiquitous across all of our collections that we did test. So since right now, our colony is all kind of rearing the same and it's all behaving the same, at this point, we're not going to form iso-female lines and break them off just yet. But we are going to continue to test our colony as well as any wasps that go through our host range testing, just to make sure we're seeing this kind of routine mix of all these haplotypes that we're expecting and making sure we don't see other things popping up in our colony. And as we continue to do future testing, we're hoping to include a wider range of collection origins throughout China, because right now our colony that we're working with is all collected from Beijing. So we just want to get an overall better understanding of like kind of the genetic makeup of these Dryaenids we're working with, and future testing is hopefully going to allow us to do that. So the next goal that we had for 2023 was to produce more females, possibly through better mating practices. So coming into 2023, we had just determined how to mate Dryaenids. And we did this through a combination of isolating males and females together in a small mating cup prior to their oviposition period. We also exposed them to some natural light, as well as excess males being put in with the females just to increase opportunities for mating. We did see that our colony was only about 25% female, which is lower than we'd hoped, but we did overall produce 119 females to do our work with in 2023. So overall in 2023, our male to female ratio is still lower than we would have liked, being only at about 32% female, but that was a slight improvement. But fortunately for us, we're able to use the cocoon length as a really solid metric for knowing whether the cocoons we're producing are going to be males and females. At almost 100% accuracy, female cocoons are going to be 7 millimeters or over, whereas males will be under 7 millimeters. So that's a really good indicators of kind of how our colony is looking at any given time. One interesting thing that we did notice, so when we look at just the colony method, which is what I described earlier with the caged nymphs and a couple wasps in there for a few days to parasitize, that did leave us with about 28% females, which is pretty consistent with what we saw the year before. However, when we look at the host range testing method, which I'll describe in more detail in a minute, but is essentially an isolated exposure of a single wasp to a single second instar nymph, that actually resulted in 70% females. So that's actually something we're going to keep an eye on and see if that's potentially a way to produce more females in the future. But overall, we did produce almost 5 times as many females this year as the year before. So this allowed us to do many things. Obviously, we have a greatly expanded colony. We were able to establish a secondary colony in Delaware with our collaborators at ARS. And it also allowed us to make significant progress moving into the host range testing in 2023. And it's going to allow us to dig into some of these life history evaluations here in 2024. And I'll just briefly touch on what a couple of those might look like. So one thing we really want to look at is really understanding the diapause of these wasps. We need to determine when the cocoons truly enter diapause and when that diapause is broken. And we also want to, so to do that, we're going to set up a study where cocoons are going to be put into natural conditions and then into chill for various amounts of time, just to figure out when in that natural cycle that diapause is initiated and terminated. And this is set to take place with our collaborators in China this year. We also want to run a longevity and fecundity study just to better overall help us understand the capabilities that this wasp might have as a parasitoid. So a couple things that we want to dig into this would just be see how many total progeny a single female could possibly produce. We do know a single female can produce over 100 cocoons, but we haven't found the maximum number of that yet. We want to see when in the lifespan of an adult female parasitism starts, peaks, and ends, and possibly whether the sex ratio of the progeny produced might also change over the course of the female's life. And then we also just have a few questions that we want to answer about possible ways to further optimize these rearing strategies, such as if there's an optimal nymphal to parasitoid density, or if we could be rearing these wasps at different temperatures to kind of get closer to that continuous colony, and so we wouldn't have to chill these wasps between every generation. But finally, our last goal for 2023 was to move into some host range testing of Dryaenus sinicus. And to do that, we'll jump right into the methods. So for this, we're only using Dryaenus sinicus females that were between 7 and 28 days old. And we just did this to avoid any of the possible observed decrease in parasitism at the very beginning of their life and the very end of their life. And these females live for about 6 weeks. So we basically just tried to cut off the very beginning and very end and use them when we thought they might be in their prime. So females and males were held together and fed on honey during this pre-oviposition period of that first week of the female's life. And then the wasp would then be exposed to spotted lanternfly nymphs from Friday to Monday before we actually started testing. And the reason that we did this was just we thought that there was a possibility that they might need the opportunity to host feed in order to potentially develop and mature any of their eggs. And then on Monday, those wasps were taken down and held with honey and water for 24 hours without any access to nymphs before their testing. We were kind of just calling this the starvation period, and this was really aimed at just hopefully increasing any of their oviposition behaviors during the actual testing. So when we get into the actual testing, a single Dryaenus sinicus female was exposed to either a non-target or a spotted lanternfly target nymph and observed in a petri dish for 1 hour. So then wasps would then be removed and the nymphs would be held on only honey and water for another 24 hours between exposures, or if it was the weekend, it would be 72 hours. But ultimately, this testing was done sequentially and in triplicates for each wasp. So a wasp was a replicate done in triplicates, so 3 exposures per wasp. And then for each non-target species we're testing, the goal is to run 30 wasp replicates in triplicates for a total of 90 exposures. So when we get into some of the results that we were looking at, just to show you some of the species that we were working with. So obviously the controls for everything was going to be the spotted lanternfly. but we also were able to test another full gourd species, Publicia fuliginosa. This was particularly important to us because it was the closest relative that we had access to the spotted lanternfly. But we also were able to test a couple of squash bugs, spine soldier bug, speckled sharpshooter, and green stink bug. And I'll say that some of these species were not necessarily high on our priority list. We didn't we did want to get a good taxonomic spread, but some of these stink bugs were more a factor of we had active colonies of these with our collaborators, and they were easy for us to get when we were ready for them. But overall, if we look at the results from the controls, the wasp was incredibly effective at attacking these controls. 113 out of 114 of these wasps did at some point during the testing attack the spotted lanternfly. And overall, if you look at the total exposures, there was about a 90% attack rate across all exposures. And then the really important part where the Anastatis did not necessarily succeed. So far, there's been absolutely zero attack on any of the non-target nymphs that we've put before these wasps, which is really exciting, especially for that Publicia hopper, which we thought had a potential to be attacked. One thing just to note is that these 3 species at the bottom, we didn't have enough resources to finish these in the last season. So these are still in progress as we get up to that 30 replicate goal. And that'll be kind of how we start this year's testing. And that's actually already underway right now. So if we just look at what went into choosing our nymphs for the target and non-target comparison, we did weigh all of our nymphs before we put them into testing. And we were actually trying to match the weights of non-targets with their controls as closely as we could. And this is kind of the breakdown of how that ended up looking. The weight range that we were really targeting was between 11 and 26 milligrams, because this is what we were weighing our second instar nymphs to be about. And we were doing this because we had observed a lot of attacks specifically on second instar spotted lanternfly nymphs. And it was really nice because this was actually able to be confirmed with our collaborators in ARS in Delaware. And so they set up a test just to look at possible different uses of different SLF instars. So they set up some choice tests where wasp was presented with a single first, second, and third instar spotted lanternfly nymph and observed for 25 minutes. And what they ended up seeing was that first instars were most commonly host fed upon, and oviposition was most common on second instars. And then those bigger third instars had the lowest rate of attack, mostly because they could fight their way out and escape. But it was nice to have our observations correlated with this research. And it showed that a second instar is a good instar to use for this host range testing, but it did also let us know that we should kind of be looking at possibly including some smaller nymphs in our host range testing just to potentially capture any host feeding of non-targets that might occur on those smaller instars as well. So if we just look at an attack breakdown, obviously we only had attack on spotted lanternfly controls, and on average this attack would occur in the first 15 minutes of observation and last for about 8 minutes, but there were outliers in both cases. We did have a wasp that didn't attack until 60 minutes, and we had another wasp that held on and attacked for 57 minutes. So there is definitely some range involved. But if we just look at the cumulative data for this, we did have 342 total exposures of Drynus sinicus to these spotted lanternfly nymphs that resulted in 307 in attacks, so about 90% attack rate. This led to the production of 196 viable cocoons. So that was about 64% of those attacks led to a cocoon. And then 138 of those cocoons were female. So that was about a 70% female proportion. And then just kind of taking a step back and looking at the bigger picture, the current spotted lanternfly distribution map in relation to kind of the distribution maps of the non-targets that we're looking at. We are trying to get a good geographic range represented here. And that's going to continue with the next species that we move into. So this list was compiled by a group of experts in the field to get both a good taxonomic range as well as geographic range represented. In a perfect world, we'd have enough resources to test all of these species right away, but we are particularly focused on getting more full gourd species tested because of their high relationship to the spotted lanternfly. However, these are also some of the hardest nymphs to field collect and raise. And so we know our collaborators have a lot of challenges ahead of them to help us get this done. But if we're looking at a little bit more realistic of what we might be able to do next year, we're going to try and focus on some more hopper species, particularly some membracids, and you can see some fulgurids in there as well. And you'll notice that those plots are kind of sparse. So it's definitely going to be a challenge to get these fulgurids field collected and tested. But we do have collaborations in place that will hopefully get us more towards that goal. And we are intentionally expanding some of our geographic ranges for these non-targets out west to focus on some West Coast species. So just some upcoming goals to finish up with. In 2023, we were able to complete testing of 3 species and start testing on another 3. We're hoping to follow that up again this year, and we were able to start slightly earlier, and we have a better network in place to better coordinate the synchrony of our wasps, nymphs, and non-targets. We do understand that this is truly a collaborative effort, and our collaborators face a lot of challenges finding especially those fulgurids for us. But we do have planned next steps in place. So we do have another year of host range testing. We're aiming to have another 6 or 7 of those species tested, focusing on some hoppers. And we're planning to incorporate a larger geographic representation, including specifically focusing out west. And we've started some early collaborations and pilot studies with some new collaborators out there, particularly UC Riverside, Washington State Department of Ag, and our satellite lab out in Salinas, California as well, to kind of help us further on this goal of continuing our host range testing. So with that, thank you. If there's any time for questions, I'll take them.
Thank you for that. It's very interesting. I'm just curious, can they attack more than one nymph? Can they lay more than one egg?
Yeah.
Oh,
So
fantastic.
a single female, we don't know the total maximum number, but a single female has produced over 100 cocoons just by herself.
Wow.
And we've also seen a single nymph be super parasitized, so they'll have 2 or even 3 thylacia on them. We don't know for sure right now if that's multiple females, each laying 1 egg, or if it's the same female, but they can be super parasitized.
Another question. That was fun talk as an entomologist. That was a blast. But kind of curious about that 15% trickle rate, and I wonder, you think there's a genetic basis to that, and can you kind of somehow exploit that in some way for rearing?
Yeah, we don't know. We don't know if there's a genetic basis for that, but like we're going to do a lot more testing, and that's going to be something we're going to be tracking more closely, is like if those all share something or not. But
you do a selection of those,
Yeah.
and mate them and see if they can
Yeah.
improve that percentage.
yeah, our genetics are very limited right now, so we do have another scientist at our lab who's going to be helping us do a lot more genetic work with these and really get a better understanding of what we're working with. Thanks.
A little bit. In terms of the survivability of the wasp in natural environments, is it suitable, or can it survive in most of the environmental conditions where we see spotted lanternfly spreading to or already established in North America?
We've not tested it in a lot of different environmental conditions. We're still working on the rearing, but it is really widespread throughout China. So like all of those different like environmental conditions, it's pretty suitable for us. So we're pretty hopeful that it'll be pretty good in this area as well.
Thank you.
So this is in relation to a previous question. So at what point is your population that you have in the lab big enough where you can start actually subdividing it and running selection experiments?
It's honestly more space and resource factor as far as that. there's a lot of resources and cages and plants and nymphs that go into this. So splitting it into like multiple lines would kind of be a little more than we could do in our quarantine facility. But numbers wise, probably do have enough right now.
Okay, so if you had a second line going that was doing a selection experiment, what would that second line be, you think? Like, what would you try and select for in that population?
It would probably be those, this is in relation to like that trickle emergence, so it'd probably be,
Yeah, also thinking about whether or not you're trying to adapt to the environment here in the United States.
yeah, so we haven't even started rearing in like US conditions. We're still like figuring out how to get like a continuous colony going, and then we'll kind of get into a lot of those like more like life history evaluations and like see how they do in various conditions. But right now we're still trying to just improve our own rearing of them so that we can have like a steady supply of them, and then we'll start seeing how they do in various temperatures and everything. Thank you.